It's also my pleasure to be here at this neighboring institute, and we have been always trying to build bridges so that we can work together, work better. Okay, so I'll be talking about application of mathematical, statistical, and a little bit of computer science models into genomic data. And this is done for various kinds of reasons. The most important is to try and relate genomic data with different kinds of diseases and health, well-being, diseases, etc. of human being. But it's also used for certain other kinds of reasons, other kinds of ways. And this is where my focus is. And I take inspiration from this Theodore Dobzhansky's statement, which is also the title of one of his papers, and which says that nothing in biology makes sense except seen in the light of evolution. And that's what we are trying to do. We're trying to use genomic data to understand evolution, to make sense of biology through our understanding of evolution. All right, just before I start, can I get a sense of the audience that I'm speaking to? I mean, how many of you have directly background in statistics, mathematics, or computer science? All right, all of them. How many of you know basic genetics and biology? Just basic could do. All right. Okay. Okay. So are you all? I mean, many of you from ISER? From this place? No. Okay. Yeah. That's okay. That's okay. Okay. So this is who doesn't know who this individual is? Well, everybody must be knowing that this individual, and this is his notebook, and he obviously was the one who gave this field a real big impetus, and it's a theory of biological evolution. It was developed by Darwin. Darwin gives huge credit, very deservedly so, but we should also appreciate that he was not the only or the first individual to do that, and he was not the first person who believed that all organic life develops from non-organic or non-life, and neither was he the first who saw that it all had some kind of a common ancestor. So there were other people. There were Greek philosophers. There was a school of philosophy in Greece who actually postulated, who actually talked and debated about organic life from non-life, and also the descent from common ancestors. And nor was he alone in publishing. The major work that he published with in terms of selection being the driving force of shaping evolution was also with Russell Wallace. Okay. But I think what really, so Russell Wallace was his colleague and his co-traveler in the Beagle ship which they took around the world. But what I think is really unappreciated or underappreciated for Darwin is that Darwin also derived enormous amount of strength and knowledge from the diversity that he observed. Though it apparently is slightly counterintuitive for many people to see that the selection, which puts a pressure on variability, which puts a pressure on diversity to expand just freely, uncontrollably, but Darwin actually got inspired and derived a lot of the power of the scientific from diversity that he observed, and he really had an eye for diversity. So just because I also anticipated that maybe you might not have many of you might not have a very deep background in biology, I'm only going to state, this is just to put things in at the same plane. I'm just going to state a little bit of things that we are going to need in order to understand the rest of the talk. Okay, so this is a cartoon of a human cell. It has something which is called a nucleus. Within the nucleus, there is this nucleus, and this is where DNA or all our DNAs are, all our chromosomes are, and all our DNA material is. There is a little bit of DNA outside of this cell, which is in these kinds of these organelles, which we have in plenty. Each cell has this in plenty. This is called the mitochondria. This mitochondria is a very small organelle. It's some kind of a remnant from an old bacteria that we all have in our cells. Our mitochondria are almost, well, I mean, there is a little bit of debate this which has cropped in recent times, but our mitochondria are essentially derived from our mothers. Our fathers generally do not contribute to our mitochondria. So our mitochondria are like the mitochondria which my mitochondria is like the mitochondria which my mom has in her cells. So this is a very small circular DNA that we have, and it's primarily maternally inherited. Most of the genetic material is somewhere within the nucleus. And the DNA is DNA which is famous for this double helical structure. Double helix is essentially like a ladder. You have a ladder, you take the ladder and hold it at the two ends and just rotate it in reverse directions. So that's what you get, that's how you get this double helical structure, and it's actually packaged in a very, very systematic way across these proteins and eventually into these chromosomes. So this is like a pair of chromosomes. The X-like looking thing is like a pair of chromosome, and all of us get one of these from our father, one of these from our mother. So the chromosomes are like paired, and we get one from our father, one from our mother. So the DNA resides in the cell. It's within the membrane-bound nucleus, as we have seen, the little kind of round thing that we saw in the picture of this cell. That's the nucleus. It resides within the nucleus, and it's double-stranded DNA. Double-stranded means this, and it's organized into the chromosomes. Okay. So I'm primarily interested in humans, and I will be primarily talking about human evolution, human diversity, human movements, human migration, and humans have 23 pairs of these chromosomes. This is, again, I mean, just to reiterate, this is a pair because we get one from our father, one from our mother. There are 22 of these, which are called autosomal pairs. And there's one pair which is essentially called a sex chromosome. So a pair of sex chromosomes. This almost entirely determine our biological sex, this pair of chromosome. So if you look at this is again a cartoon. So this is like chromosome 1, chromosome 2, etc., etc. This is a specific phase at which if you put a certain kind of chemical in the cell, so this is how the chromosomes arrange them. And the naming of the chromosome is pretty simple. It's the longest one under the microscope, which was named as chromosome 1, or the pair of chromosome 1. So this is chromosome 1, it's chromosome 2. Now it turned out that chromosome 2 is slightly longer than chromosome 1, but however, I mean, it was deceptive. So 1, 2, 3, and so on and so forth, it actually decreases in length. So the 22nd chromosome for humans is the smallest chromosome. The sex chromosome pair is here, as you can see. I mean, it's drawn like this, and it's asymmetric because the sex chromosome contains a X and a Y for males, and an X and an X for females. So the X chromosome is long. It's almost like chromosome 2, and the Y chromosome is the smallest one. So males compared to females have slightly less DNA material, and just slightly less. And your father has contributed a little less compared to your mother, okay, into your DNA material. Yeah, this is okay. So the building blocks for the chromosomes are nucleotides or bases, which has these names, but we call this A, T, G, and C. Just knowing this is enough. So this is like an alphabet. The alphabet consists only 4 letters, A, T, G, and C. Within a DNA, within a double helix, the A always pairs with a T and the G always pairs with a C. So this made life a lot simple for us, which means that we really do not need to know both these bases on a chromosome. Just knowing one gives a redundancy because the other one is automatically determined by that, right? So knowing just one of this is pretty good enough. Which gives us also the ability to look at this as sequences of integers, sequences of A, T, G, and C. So we can essentially look into one chromosome. We can essentially look into one chromosome as a linear sequence of A, T, G, and C. All right. So the challenge really comes because in this 46 chromosomes, which are wonderfully organized, there are about 3 billion of these base pairs. So that number is essentially where essentially the challenge comes in. And I mean, this is an aside. I do not work on this, but it's just intriguing to me that just think about this. It's 3 billion bases, and if you put them together in a linear fashion, it's about, I do not know, I think it's about 3 meters, right? It's about 1 meter, or a little less than a meter maybe. And we cannot see a cell by our naked eyes. A cell is microscopic. Within the cell, the nucleus is even smaller. So this thing is packaged in such a way that all of these 3-4 feet actually goes pretty well packed within this nucleus. And not only that, the most important thing is that every time we ask for something, like I'm talking, and which means that there are specific kinds of proteins which are being produced in my body because there is a specific kind of reaction which is going on, or when I was eating, there is a separate kind of thing which was going on, which means that it's calling for different kinds of things to be produced. And all of this, which is wonderfully packed in here, is not only packed there, it's also a wonderful information retrieval system because it retrieves, it goes to the right place and gets the right protein out of it just like that. And it really takes no time in doing that. Otherwise, we would have been slow in responding to anything which is like a change in our environment, right? It really doesn't take a lot to respond to a change to an environment. But just imagine, any change to an environment would mean that something else needs to be picked up from here. And it's all packed into this compact thing. So this packing and unpacking is just a fascinating thing. The other thing which makes life easier for us is that DNA for any 2 individual is pretty similar. It's about 99.9% identical. It depends a little bit about, the numbers vary a little bit about depending on whom you are talking to. But roughly, on an average, any 2 individuals sampled from this world are different in 1 in 1,000 places, which is advantageous. So what that means is we do not need to deal with these 3 billion base pairs all at the same go. What suffices is 1,000th of it, which is about 3 million. So if you're comparing between 2 individuals, there are only about 3 million differences. I mean, 3 million is a huge number anyways, but compared to 3 billion, it still is handleable. So the problem, however, is mathematically much more challenging because what we find is that in order to do all kinds of things that we do, it's not this differences in 3 million, which is only what is important. Of course, that's important, and that's the key thing. But it's also important because there are many kinds of combinations which you can make out of this. So it's essentially a combinatorial problem. So whenever in evolutionary time point, whenever there is something new kind of a functionality which is assigned to a a living organism, it's not necessarily that the will is rediscovered. It's oftentimes whatever you have, the bits and pieces, the building blocks, with that you organize and reorganize and make it a combinatorial issue so that you can address that. Okay, so that's how it works. I also like to introduce. Well, I'm pretty sure it's not a real introduction, but there are a few words which I'll be using, and when I use it, I might miss it if there's even one individual who hasn't understood that. So I'll talk about mutation as a verb, which is like a permanent alteration or change in this nucleotide sequence. So these nucleotide sequences, as we have seen for any two individual, like you and me, or any two individuals sampled from any two places in the world, are essentially same except for 1 in 1,000 places. But if you ask the question that why is it that there is differences, why is it that there are even these few differences? So one of the major reason, or the real primary driving force behind all of this somehow goes back to a permanent change, alteration in these nucleotide sequences. These changes are more or less random, and because it's more or less random, we can apply our mathematical and statistical probability models with this. So just imagine that this is a sequence. As we can see, that there are these 4 letters A, T, C, and G, and this is a sequence, a stretch of sequence, and at some point of time, maybe this changes to this. So when does it happen? This happens primarily when cells divide, and all of our cells divide. That does not impact too much because it divides and gets into one cell and it gets into another cell maybe, and generally when the cells die off, these mutations also die off with them. If you expose an individual to what we call carcinogens or external insults, oftentimes the rate of mutations change. So you might be knowing that people say that if you are exposed too much to ultraviolet light, then these kinds of changes happen, or if somebody exposes somebody to smoking, then these kind of changes occur much more frequently. And some of these, in most cases, it will be just innocuous and will die its natural death. Some of these, sometimes when it accumulates on genomes, it can give rise to dangerous diseases like cancer, etc. When does it get important? When it gets important is during during reproduction, there are these sperms and these eggs. So if any of this happens in the sperm and the egg, then what happens is that the progeny gets a nucleotide which is different from the father or the mother, right? So that's when it really becomes different. Again, I mean, just because it's being copied, there are differences, and that's why if you compare your genome with your father's and your mother's, there are certain random places where you'll be different from your father, both your father and your mother, and that's because of just these random changes that has occurred in your genome. All right, so this is one thing which is going to be important, and as you understand, obviously that this is one of these primary things which we try and model, and this is which we observe and which we try and model. And the other thing which also is very, very important is something which happens in not every organism, but in organisms which are like us, which we call our diploids, because we get this chromosome sisters from our parents. So what this is called is something. What happens here is something like this. That suppose you have this pair. Okay. Suppose you have this and this and let me take this. This one, which is a complete red, and this one, which is a complete blue, and at this region. So they are pretty much the same. Its organization is same. The sequences are pretty much same. sometimes it breaks from here, and this red part which breaks from here can get stuck here, and the blue part, correspondingly, blue part breaks from here and gets stuck here. So if you have a chromosome like this, it sometimes breaks at regions, and then it gets exchanged. So this is called recombination, and it happens between two sister chromatids. So just to put this in perspective, in a visual, so that you get a sense of this in a very simplified kind of a model. So this is not necessarily our ancestor. This is ancestors to us and chimpanzee. So we and chimpanzee, we have a common ancestor sometimes back. So suppose the sequence of that ancestor at a specific region, as a specific small region in the genome, is this yellow sequence. I mean, however it is. At this particular locus, at this particular position on the genome, let the nucleotide be an A, right? So our common ancestor, our chimpanzee and our common ancestor at this particular location has an A. Now suppose this A to G transition is like a mutation. So suppose this has happened somewhere back in time, and this somehow survived in the population, which means that this is here and it's passed on to individuals, and that is passed on to even individuals, and eventually both of these are present in the population. Similarly, consider, so at some point of time there are these A here and G here on the chromosome. So some individuals carry this, some individuals are carrying this. I'm looking at another position where there is a T and a T. Everybody is same. Here is a T. And suppose there is again a change, a random change, a mutation to this T, and it becomes a C. So now, if you look into this, this has happened on the background of a G. All right. So there was a AT chromosome, which was the original chromosome which our ancestors all had. Then there is this chromosome which was GT. That was because a change here has happened. And then a change here has happened and given rise to this new one, which is a GC.
So you are considering one pair of sequences.
So just now imagine the I've just broken down the sequences as just separate. So it will
be in pair 80 and 70.
Yeah. So you can imagine that this is essentially, there is another yellow here, right? No, it will not change. So if there is a change here, which is on one chromosome, it will not change. What will happen is, if you think about the DNA helix, that if you change the A, the corresponding thing with which it's paired in, that changes. But this is something which suppose I have got from my father, and there is something which I have got from my mother. That does not mean if there is a change in one of this, that there will be a corresponding change in the other. That's not true. But within the helix, to maintain that structure and to maintain the integrity of that, if one of them changes, the other one changes immediately. Okay, all right. So this is like breaking the chromosomes and just looking at it as a pool, not like looking at this as chromosomes within an individual. So this is the ancestral one. This is one which has changed to G, and this is what you had in the pool. Just imagine in the bucket you had this, and then at another position, which is somewhere down the line, you have a T going to C. So now you have three of these different kinds of chromosomes, right? A AT, a GT, and a GC. So this was the ancestral. This is the one which you got from this particular mutation, and this is the one which you got from this particular mutation, right? And now, if there is a recombination event which happens somewhere in between this and this, then it will exchange materials. So if this yellow and this blue gets recombined, then it will be like a break here and A and C. So this will, if there is a break here, this will get here, right? And you will get the GT, which you already have. But this will give rise to a new kind of a combination, which is an AC. Is that, do all of you see this? Okay, let me try and show it again. So just imagine that this yellow thing, which is a long stretch, which has an AT, right? And then there's this blue thing, which is a GC. And what happens in recombination is it breaks at one of these positions. Suppose it breaks here, and this material is exchanged. which means that it gives rise to A, and from this breakpoint, this entire thing gets here. So this part is a C. So this gives rise to this new combination. Whereas this part breaks and gets stuck into this one. So this is a G here and the yellow T here. But this already exists in the population. This you have already got from previous mutations. So this gives rise to a new combination. Now, if you just imagine that there are two positions here, two possibilities here and two possibilities here, so there are only 4 possibilities possible, right? I mean, and the fourth one generally is not because there is a mutation happening somewhere. So these combinations arise because of mutations here and mutations here. The fourth combination generally appears because of recombinations between these existing combinations.
Sir, the corresponding pair will not change. You said
The other pair, so yeah, so just imagine, so this is like this and this, right? So just imagine the green one is not here. The yellow one and the blue one is here. And the breakpoint is right here. So what happens is this block gets exchanged with this block, which gives rise to this one. This is a new chromosome which it gives rise. But the other part is going to be G here, and from here you get this T thing, right? So that already exists in the population. So that will not give rise to a new combination. That will give rise to a new chromosome, but not a new combination. This combination already exists.
and the bond will exist between them if it will go into mutation like
The-
this.
So
Means if it will go mutation like this, the bond between the pair AT and C is there. But after the mutation, in place of A, it will come C.
okay,
So the bond between the pair.
let me try and illustrate this. So this is like this, that the AT bonding is like this. All right, so this is within a chromosome, and these are the bonds. This is, if you choose this, this one automatically gets selected. So suppose this is what our original chromosome was. All right, and I'm looking into this and this. These two places. There are sequences in between, but I'm just looking into these two places. So every individual is supposed like this. Then at some point of time, there is a mutation here which makes this. So there are many copies of this, right? There are many people who are like this. There are many copies of this. Some individual gets a change here, which means that this thing changes to a G. So this becomes a G, this becomes this becomes a C. So there are still many copies of this thing in the population. There are many copies of this in the population, then there are some copies of this. Now, if this increases in proportion, then there will be some proportion of this in the population as well at some point of time, after a few generations. This is not happening in one generation. This is after a few generations that you have a few copies of this. All right. Oh, okay. So then consider this particular. Then consider this particular locus. It's a different locus. All right. This is a different locus. So every individual is like this because people are similar. At this locus, on this background, there is a mutation which makes this into a C. So this changes automatically to a T. So there are still many people who are GT here. There is a new combination which is a GC here, and the old combination still exists. So all of this exists, and this is the new thing which we get from a mutation here.
That will be CG.
Yeah. CG. That will be a G. Yes. So after this change, this will automatically change to a G. And but the fourth combination generally also, so there is also a fourth combination which you will eventually get, which is a here and a C here. And how do you get a fourth combination? Either you get a fourth combination by changing this to a C, which is a very low probability even because there are so many places, there are 3 billion places and there is very little chance of that thing happening here again. What happens much more frequently is that there is a break between this, and this part and this part gets exchanged. So that's how you get all of these combinations.
For this material to pass on to the next microsome, the mutations has to occur in the sperm or the egg?
Yes, yes. It has to occur in the egg or the sperm, otherwise obviously it cannot pass. All right. So the major things that we find are that there are 3 billion bases, and there is a lot of dependencies in the sense that most people are anyway similar. So if individuals are anyway similar, and there are, if you are comparing any two individuals, there are only 1 in 1,000 differences. There is merit in finding or in making one reference sequence for all individuals, and that was a project which was undertaken, and it was called the Human Genome Project. It was planned for 15 years. It was initiated in 1990, and just to imagine the enormity of this project, the way it was done is, so just imagine that you have 3 billion bases, all right? So when this project was undertaken, people had the technology to, at one go, sequence 500 bases, all right? So you do not know what you would do. You just had a few random starts. So so this is, suppose, all 3 billion bases. This is all in chromosome 1. You have put that in with all in chromosome 2 and so on and so forth. So suppose this is your 3 billion bases, and people could only, from random starts, they could only sequence 500 bases. So this was the state of the art when people started doing that. So, that too is not like that you start from here, do 500 bases, and then start from here and do 500 bases. They could only do it from random start points on this genome. So, but they were ambitious enough to take a project like this, and what they envisioned to do is something like this. So if you get a random start from here, you do 500 bases, and if you get this 500 bases and get a few random starts, then from here you design a start within here and get 500 more bases. And like this, you do this with overlaps and eventually span this 3 million. So just imagine how many times you have to do this. So obviously this was planned for 15 years. It was initiated in 1990. The first draft was published in 10 years, in 2000. It was supposed to be completed in 13 years, which is two years less than when it was estimated or anticipated. And that was because there were many, many new kinds of technologies which people have invented during the course of all of this. People said that this is an achievement bigger than scientific achievement, bigger than humans landing to the moon. And the cost of this was only about 2.7 billion USDs. So which means that it's almost like $1 US was spent to generate one single base on the genome. And this involved all the great universities and all the greatest minds in the world to work together to do this. So it was a huge achievement. It had huge amount of man hours spent in just doing the sequencing. And then there was this wonderful methods by which people put all of this together and stitch this into one single genome. All right, so there were a few things which are interesting, and if you go back and do the calculation, you can all see this, that though it just sounds enormous, but on this 3 billion bases, if you just take a random stretch of 25 bases, that's pretty much unique. All right. So there are these 4 combinations possible. So there are 4 raised to the power 25 possibilities if you just take a stretch of 25 bases. But on this genome, that's so large, that number is so large, that on this genome, a random stretch of 25 bases is pretty much unique. So if you just could identify a region where it's common in 25 bases between any 2 of these 500 stretches, you know that there is an overlap between them. It's pretty much, it's almost true for all over the genome, except for very few places where there are specific kind of structural variations. So this was again a great advantage, and that's how it was done. So this is actually a snapshot from real data, and this is how data that we handle actually looks like. So as you imagine that that in most places, because individuals are the same, this is one of the regions where we see mutations more frequently. We have chosen one of these regions because otherwise there will be only one change in 1,000 places. So this is like one individual, right? You and my data actually looks like this. In most places, we don't need to even look at what this is, because our father and mother are like any 2 randomly chosen individual from this world. They are also very, very similar in their genomic sequences, except for 1 in 1,000 places. So in most places, the chromosome readout that I get from my father and the chromosome readout that I get from my mother is essentially the same. And that is actually same as the reference sequence. All right? So that's the merit of having a reference sequence. So all of this, even without mention, you know that that's going to be like the reference sequence. In some of these places, this individual's father and mother were different. Suppose I'm concentrating on this. Here, one of the parents had a G, another parent has an A, right? Of the A, T, G, and C possible, one parent has a G, another parent has an A. So this individual's genome at this particular region is a readout of GA. And one of them you can put your bet on is also either the G or the A is the one which you find in the reference sequence. So this individual is for every position, this individual is called what we call is a homozygote, or this is same, or both the zygotes of this individual were same or had the same DNA. So this individual is homozygote. In these regions, this individual is called a heterozygote. All right, so this is a typical genome which looks pretty much like this, except for a few places where there are these kinds of heterozygosities. So this genome is our data, and I'll just quickly go through these definitions. Again, a gene, this is only a conceptual idea of a gene, which is a segment of DNA contains information producing a protein. This was conceived way before people had any idea about DNA. This was, people just observed individuals and saw that individuals resemble their relatives and thought that there might be something which is passed on from one generation to another, and they called it gene. That material they conceptualized as gene, and that we now know is a segment of DNA which gets passed on. The variant forms at a position is essentially the alleles. So if you just go back to this, you can also imagine this as 2 alleles, a G allele and an A allele. We can also think about it in this way. And then there is a locus. Because it's fixed, it's almost like you can have a starting point from where you can count. You can go and have this as a position. This is a position. This is a specific position on a genome. So having a reference sequence gives you this power that you can say that on chromosome 1, position number 1, 2, 3, 7, 8, what is the nucleotide that you observe? So it will give you a nucleotide, say A or T or G or C. Okay, so So what does genes do? Genes do many kinds of things. It's responsible for many diseases. This is one of these unfortunate diseases, which is known as Down syndrome, which gives which happens is what happens is the chromosome 21. There is an excess copy of chromosome 21. So generally, what people think is that if you do not have something, or if something is not working, then you generally have diseases. It's not always true. Sometimes if something is there in excess, you can still have a disease or a condition. This is Down syndrome. It results in a wide range of developmental delays and physical disabilities, but it gives rise to very, very nice and happy people. These people are generally very nice. But it's not always genetic. So if you clasp your hand together like this, then all of you will see that one of them, either putting the right on the left or putting the left on the right, is actually a little bit more comfortable. And people have looked at this in families and found that even something as innocuous like this has a very strong genetic component. You can go and ask your parents, and in most cases, you'll find that your comfort and your parents' comfort are pretty similar. Rolling your tongue, again, extremely innocuous, but probably gives nothing. I mean, so these are examples of things which are controlled by genomics, which have even a functionality, a pretty strong functionality, but has nothing to do with selection. This does not give an individual any kind of an advantage in terms of how Darwin has looked at this, and it does not give an individual a reproductive advantage, right? But it just gives rise to different kinds of variations in the population. So some of you will find that this is very easy. Some of you will find this to be extremely difficult. All right, so yeah, I mean, this kinds of earlobes. Okay. So all of this is to give you a sense of how this is connected to different kinds of functions, different kinds of biological functions that we do. But meanwhile, what is also done is using this DNA as a book of history, and that's what will be the major part of my talk now. So how do we actually use this? So the intuition is pretty simple, which is that the early idea of evolution had a concept of a common ancestor, right? If there is a common ancestor, then the common ancestor is ancestral to many of us. If you take two individuals who are relatives, they have a common ancestor not far back. If two individuals are distant, they have a common ancestor which goes back a long distance. And the whole evolutionary paradigm is to have common ancestors even for species, right? So if you consider two closely related species like humans and chimpanzees, people say that they have a common ancestral species. The species might not be living today, but there was a common ancestral species somewhere who's common ancestor to both humans and chimpanzees. All right, so what we do is that if you have common ancestry and you have variation, many of these variation are because of this dynamic molecule called your DNA, where there are changes which are occurring. Maybe there are timestamps using which are here, and maybe we can decipher these timestamps using the kind of data that we have. So the whole premise of population genetics is what processes might give rise to the observed patterns of variation. So this is essentially, to many people, this will immediately sound like a Bayesian framework, where you have some kind of an observation and you're trying to fit in a model, or trying to fit in history, which should give rise to a variation which is like this. Okay. So why is that even possible? So this was first observed by this lady in 1963, and what she was studying was not essentially DNA. She was looking at proteins, and she was looking into similar proteins in different species. All right, and what she found was that if you consider two species whom we consider are similar, say, for instance, if you consider humans and chimpanzees, and if you're looking into the cytochrome C of humans and chimpanzees, then their cytochrome C will also look similar. So all the protein, well, I mean, many of these proteins, which you consider when you're comparing between different species, the proteins are also similar, and it's apparently she was intrigued by the correlation of the distance between this and the species itself. So what later people have used this is something like this. So this, in this graph, the x-axis are just different species. Here are baboons, here are cows, quail, chicken, alligator, so on and so forth. All right. The y-axis is one kind of a distance. The x-axis, the y-axis here is another kind of a distance. But what they're looking at is the field circles are fossil divergence time, which means that when is the earliest evidence of a baboon that you can find, all right? Which means that suppose this is for cows, suppose it's 80 million years ago, right? That's fossil evidence, fossilized evidence of the first something which resembles a cow, and that's about 80 million years ago. Similarly, you can do it for different species, and this was what paleontologists would do, and they had this whole set of fossils which they dated using different kinds of methods, and they had timelines for all of them. Now, what post-Margolish, what people have done is they also looked at their molecular divergence time, which essentially means that they have looked into their proteins, many of these proteins, and looked at the difference between proteins. And then they had a conversion factor by which they multiplied that. And when you convert that, what you find is that the molecular divergence times estimated from the difference in protein is very similar to the fossil divergence time. Now, which would mean that it's apparently that these proteins started to diverge at the time this particular species appeared on this world, right? Generally, if you compare between them, you'll see that generally, except for chickens and alligators here, generally the molecular divergence time is slightly older. All right, the fossil divergence time is slightly younger. So this is present time. This is going backwards in time. If you look at the fossil divergence time, that is slightly younger compared to the molecular divergence time. And that's also pretty intuitive, because you can always, in the future, discover a fossil which is older than this. So this is the oldest shark fossil that people have found as of today, right? Someday later, you can find a shark fossil which is even older, and it can actually close down this gap. And surprisingly, that is what has happened over the course of time. So when these molecular methods first started, these molecular methods are much more recent compared to the archaeological records, of course. Archaeology and paleontology has been dealing with this for a very long period of time. They have all of these calibrated, and all our textbooks were written all according to this. The evolution textbooks were all written according to this understanding, right? So molecular biology came later. And when it came later, it had to actually corroborate its findings with the findings of the existing disciplines, right? And that's what started happening. You find this strong correlation, and not only do you find correlation, you also find that over time, for these disciplines, sometimes they started getting fossils which are actually closer to this estimate. The great thing about molecular divergence is that you don't even need this, right? You don't even need the uncertainty of whether this is, the uncertainty to answer that whether you have actually got to the oldest fossil. You will never get the oldest fossil, right? That's almost sure for any species. They will never get the oldest fossils, but you can still estimate the time frame with if this is true. Now, why do this hold? why does it hold? It holds because the rate at which the mutations accumulate in species is somehow scalable, is somehow constant. If that was extremely random in one species compared to another, then you could have never got this. Because we haven't observed this. We just have a notion of that if you have X differences between these two, that calibrates to a Y difference in terms of their ancestry or common ancestor. So if that, because that can be done, as this shows, that probably that can be done, which gives a consistency in the molecular clock, which also gives us the whole power of dealing with all of this and try and figure out history, history and prehistory of this. This is much more to the molecular period. This is 1975. And this is one paper which came up in Science, and this is one which actually took the world by storms because of various kinds of social issues. So people looked into humans and chimpanzees, and this individual, many of you might be knowing that this individual, Mary Claire King, she is famous because she found the genes which predisposed, one of the genes which predisposed to a very specific kind of familial breast cancer. So Mary Claire King's early work was in population genetics, and what she and Alan Wilson, I'll talk about Alan Wilson when I'll narrate stories about human evolution and how we have used genomic data to find that. This is her graduate work, and she looked at human and chimpanzee sequences and looked at differences between them, and found that the genetic material, if you compare the human and chimpanzee sequences, they are extremely similar. They are around 99.5% similar, and that took the world by a storm. I mean, people never believed that we can be that close to chimpanzee, that we are different in only 5 out of 1,000 places. But I mean, also they postulated that genetic distance between human and chimpanzee is probably too small to account for their differences in the organic level. So, and that is what is also holds true, that you do not really need to rediscover the wheel whenever you want to define something which is a new function. Okay, so the basic premise is something as simple as a distance matrix like this. So suppose you have sequences H1, H2, H3, H4, H5, H6. What you do is you just count the number of differences here, right? So H1 versus H2, there is only, if you compare between them, there is only one place where they're different, possibly at this position. If you compare between H1 and H6, or H5 and H6, then there are H1 and H6, then there are 5 places where they're different. All right? So just by this data set, if all of them have a common ancestor, then obviously H1 and H2 are closely related, whereas H1 and H6 are much distantly related. And this was the first way in which obviously people all started doing this. Even if you think about this, this probably comes intuitively to the human mind, because if you look into Darwin's notebooks, Darwin also actually drew trees like this, right? If you just remember the Darwin's notebook that I also had here in my slide, Darwin was trying to draw trees, trying to connect relationships between species by drawing tree-like relationships between them. All right, so that was nice, having a tree-like relationship depending on the distances that you have. But what happened later is that harnessing this whole understanding that people might have common ancestors, there was very elegant models which were built, and the most important of them probably is the coalescent model. So this is not an exact theoretical. What I'm going to show in the next few slides is not exactly the theoretical way in which the coalescent model is built, but this will probably give you a sense of how you look at it. All right, so time here is present. All right, this is today. Here is present time, and this is past. Time is moving in this direction. The advantage of doing this is that you can actually collect individuals here, all right? So suppose there are 22 individuals who are from this present generation. And if you go one generation back, this is a simplified kind of a thing. If you go one generation back, what will happen is some of these individuals will have common ancestors, right? So if you just go one generation back, people might have, people might not have common ancestors. So here, every individual have separate ancestors, whereas these two individuals are like siblings today, they had a common ancestor here. These two individuals have a common ancestor. All right, so there are some pairs who have a common ancestor. Now, if this is a premise that as you go back, you can have common ancestors, then your number of independent individuals in the previous generation is only going to decrease, right? So you start with 22 individuals, some of these individuals will have common ancestors. So in the previous generation, you have only 18 individuals left here. If you started with 22 and if you had 22 in the previous generation as well, then if you have common ancestors, then the number of individuals is only going to decrease as you go farther and farther back in time. So you have from 22 to 18 to 16 to 14, 12, 9, whatever. Eventually you get to your single common ancestor, right? And what happens after that is you can actually trim everybody else out. So this is your single common ancestor. So all of these 22 individuals eventually, some way or the other, are related to the single common ancestor. All right? And if you now trim it completely, if you trim everybody out, then this is what you're left with. And this is called the most recent common ancestor, or the MRCA in the literature. And the whole idea is to have these individuals going back to this. Now just imagine that, that if you have sampled individuals, or if we just sample individuals who are apparently unrelated from a population, like here, then we will, as we go back in time, we'll have common ancestors somewhere. Now this will give rise to different kinds of relationships, phylogenetic relationships between these individuals and this, right? And to give a sense of how this will, so if you just have three individuals, A, B, and C, who are distinct, then you can have three different kinds of topologies if you consider A, B, and C as distinct, right? Because I mean, even with the most simple cases where you do not, so this is just one kind of tree that you can have. And these labels, if you change these labels, you can have three trees just because you can change the labels A, B, and C. I mean, there is also a theoretical possibility of a tree like this, but that we are right now, we're just looking at bifurcating trees and not a combination like this, because this will give rise to even more possibilities. We can show that, well, I mean, for coalescent theory, this particular one is not terribly important. So with three individuals, there are three kinds of distinct trees, and as expected, if you just increase the number of individuals, it just exponentially increases, and it becomes almost a non-trackable problem only with 40 or 50 or 100 individuals, right? So the problem is that it's almost impossible to do the sum over all the possible genealogies. It's just too big a space to do all of this. So is there a way in which we can trim it down? And the answer to that is, of course, there is. The simple way of looking at it is, so suppose you have sampled 10 individuals. So this is 10 individuals, and these are just a sample of few trees which are here. The most important problem is that there is always a random variable which is on the y-axis, which is the time to the common ancestor, because that is something which is always a probabilistic thing, right? And then there are these different kinds of trees which are possible, tree topologies which are possible. So the way in which we deal with this is something like this. All of this happens independent of any mutations happening on them. But just imagine if you can track that this is a common ancestor, which means that this individual has some kind of DNA sequence. And at this point of time, from this generation to the next generation, suppose there is a change in the G to a T, right? This is of course a very simplified kind of a model. Then what will happen is all the descendants of this individual will have T here, whereas everybody else will have a G here. So if we sprinkle mutations happening on this tree, then we can get a better sense of the tree itself. So if you have here, you immediately know that these three individuals are probably much closer compared to their common ancestry, whereas these individuals and these individuals are much different when you compare this group with this group, or this group with this group with this group. So that is what we do, that we add the mutational events, are not added to the genealogical tree. The way it essentially done is slightly on the reverse. We look at the mutation which we have, observe the mutation, and try and reconstruct the tree using that, but that becomes possible. So just imagine this, that this is the most common ancestor. This is the most recent common ancestor of all of these 22 individuals that we had here. And suppose this is the sequence of that individual, right? So if there is no mutation, everybody will have the same sequence. And you will not know how all of these 22 individuals are genetically connected, or what is the phylogenetic relationship between all of these individuals, and how do they coalesce to this individual. But just imagine that suppose there is a mutation here from a G to a T. So a mutation has happened here, which gives rise to this red stretch of DNA, which means that if the mutation has happened here, this individual is common ancestor to many individuals here. So every individual who is a descendant of this individual will have the same sequence. So this will immediately define a closeness and distance in terms of the mutation on different individuals. Suppose there is a mutation on this branch right here, and these individuals get this kind of a color. So you can go on adding these mutations at different places, and then you can get all of this. And if you get this, then it becomes much more easier to reconstruct this under certain kinds of assumptions. Okay. Okay, so the other thing which has come to our advantage is that these mutations which arise in different time points do not necessarily confer our selective advantage, which means if somebody has one of these mutations which arise, that does not necessarily give that individual a selective advantage to have more offsprings in the next generation. That means that all of this can be handled much more in a mathematical way, and you do not need to impose different kinds of restrictions and different kinds of functions whenever there is a mutation arising. So majority of these variations are just neutral polymorphisms. These are called neutral because it does not confer you advantage in terms of reproductive fitness. There are some which under certain conditions do, but in most cases it does not. So if you consider, if you not just consider something which is as simple as that there is a population which has a size of capital N, all right? So the population has a size of capital N, and you are suppose just taken 2 individuals, all right? Or if you think about a population of size capital N, because there are n individuals, then every individual is diploid, so there are actually 2 n chromosomes. Then if you just take any two individual, the probability that these 2 individuals will have a common ancestor in the previous generation is that of this 1 upon 2n, they have to choose only one individual, right? They have to choose one of these 2n possible individuals or chromosomes in the previous generation, right? Is that clear? So there are n individuals. It's a constant population which means it has remained constant over time. N individuals means 2n chromosomes, and if you just take 2 chromosomes, these 2 chromosomes is derived from a common ancestor in the previous generation with a probability of 1 upon capital 2n. Right? Just because there are capital 2n possibilities and because there is no selective advantage, the chances that you get this is, it's equally probable whom you choose of this, all this 2 of capital N individuals. So the probability of this not happening is 1 minus of this thing, right? So okay. So this is something like this, that so here are my so this is suppose the whole population. So this is individual 1, this is another individual, so on and so forth. This is another individual. So there are n individuals, which means there are 2n of these chromosomes. So you are apparently looking like you have put all the chromosomes in one bucket and you're looking back in time. And if the population has remained constant over time, then in the previous generation as well, in this generation there were 2n chromosomes, in the previous generation as well there are 2n chromosomes. Right? Now the probability of having 2 individuals having a common ancestor is that both of them choose the same individual, which is nothing but 1 upon 2n. That's the probability of this happening. So the probability that 2 of these chromosomes will have a common ancestor event is 1 upon 2n. And that not happening is 1 minus 1 upon of 2n. Right? Now, if I now ask the question that what is the probability that the first common ancestral event happened t plus 1 generations back. So this would mean that for the first t generations, there was no common ancestral event happening. So this was the probability of that raised to the power t. So this is essentially like putting this into a binomial, into a negative binomial kind of a distribution. This is the probability of success. This is the probability of a common ancestral event. And this is the probability of a failure, which is not having a common ancestral event. This has happened for t generations. This is the first time you're getting success. So that probability is 1 upon 2, 1 by 1 upon 2n raised to the power t. Now, so for a population which is constant in size, if you have sampled 2 chromosomes, then it's just as simple as doing this. Now, instead, if you have sampled n chromosomes, or if you have sampled a small n number of individuals from capital N number of individuals, obviously it will change the numerator, because you can choose these 2 chromosomes at small n choose 2 ways, and this is obviously also going to change the numerator. But it still remains like a negative binomial kind of a distribution. All right. Okay. So for the time being, if you consider that this is just like a haploid individual. All right. So I'll just make a small, so suppose you have just, let's say 5 individuals, all right? So this is 1, 2, 3, 4, and 5. And this is a population which has remained constant over time. So of this, these individuals have the first common ancestral event, suppose T5 generations back. So as you can see, this is a random variable which follows a negative binomial distribution, right? This is a random variable which follows negative binomial. So I have sampled 5 individuals in a population of size capital N, and T5 is the time when I get the first common ancestral event. But now if you think about this, these individuals have distinct ancestors here, right? So these individuals have distinct ancestors here, and these 2 individuals now have a common ancestor here, which is equivalent to actually sampling 4 individuals from this time point. So this is present time, and this is like sampling 4 individuals from this time point. Now suppose these 2 individuals are coalescing here, and I call this as the second random variable of the time taken for these 4 individuals to coalesce as T4, which again gives me this handle that this is like sampling 3 individuals from this time point. So suppose now these 2 individuals coalesce, and this is T3, and eventually, which is like sampling 2 individuals, which is T2. So essentially, the time to the, if you have 5 individuals and the time to the most recent common ancestor, essentially is T2 plus T3 plus T4 plus T5. So this is just adding over in a generalized setup. If you have T, or the time to the most recent common ancestor, that's just T1 plus T2 plus so on up to Tn if you have sampled little n individuals from capital N. So if you I mean, this applies absolutely directly to colonies of bacteria, where generations are essentially distinct and can be looked at discrete generations. It also works pretty well for humans, and it can also be shown that if you use a diffusion kind of an approximation where these generations are not necessarily non-overlapping, you can still get to pretty much similar kinds of results. Now the fascinating thing is that if this is the total length, if you do the algebra, you will find if this is the total length, which is a random variable, of course, because it's sum of this n, the sum of this, actually this is T2, starts with T2, sum of these n minus 1 random variables. If you have n, little n individuals whom you have sampled, it's sum of n minus 1 random variables. This entire length, if you consider the expected length of this tree, then the expected length of this tree is only double the time it has taken from here to here, from 2 to the most recent common ancestor. So if you just do the algebra, it's actually, it's simple. It sounds a little counterintuitive, but it actually is that the time taken, the expected length of this tree is only double the time it takes for 2 individuals to coalesce to a common ancestor. Okay, so this is how it looks like. So these are random variables. If you go back this, you can add this up, and this gives you a pretty good mathematical handle in looking at this. The other thing is that it's also easy to see that, just imagine that this population in the past has been increasing in size, which would mean that in the previous generation you will have a population size which is n1, right? In the previous generation you will have a population size which is n1, which is smaller than this capital N, which means that the probability of finding a common ancestor actually increases. So this tree will shrink in length if this population is increasing in size, because every time this probability increases, and the expected time to this common ancestral event will decrease at every step, and for a population which has been increasing from past to present, this tree will shrink. It will be exactly the reverse if you see have a population which is shrinking in size. Because every time you go back in time, there are more options of common ancestors which are available now. All right, so this coalescent has given us a very good handle to handle things like migration, expansions, bottlenecks, and selection. So bottlenecks are things which has happened in history of species many, many times, which essentially means that population sizes have suddenly shrunk due to different kinds of reasons, and then eventually they have expanded. And all of this, this also gives you very, very efficient programs by which you can handle all of this. So now you can have programs by which you can sample individuals from populations, you can generate these trees, you can have a mutation rate, you can sprinkle the mutations on these trees and see what patterns give rise under what kinds of demographic scenarios. So you can change the demographic scenarios according to your will and see what kind of patterns you are expected to observe, what kind of patterns are observed if you change the demographic scenarios. All right, so are you taking a break now? Okay. Okay. So I'll talk about a few things which has been derived using this kinds of methods. Before doing that, just imagine that we were talking about mitochondria. So our mitochondria are material which are derived directly from our mothers. So if you think about mitochondria, then the common ancestor theme is exactly directly applicable to a scenario where you have sampled mitochondria. Because if you have sampled mitochondria in this generation, right in the previous generation, it will go back to your mothers, right? Everybody who has been sampled, irrespective of the biological sex of that individual, goes back to the individual's mothers. And from there, it can only go and coalesce to their mothers and so on and so forth. So just after one generation, it's all females in the population, and then it goes back and tracks the female lineage. Almost similar thing can be said when you're sampling the Y chromosome. So if you're sampling the Y chromosome from this time point, you can only sample the Y chromosome from the males, and the male's Y chromosome is derived from their fathers, and that there is from their fathers, and so on and so forth. So that gives you a lineage which tracks back the male lineage of a population. So if you look at the mitochondria, it looks into the female lineage. If you look at the Y chromosome, it looks into the male lineage of the population. And it does not have any kind of complication in terms of recombination, which means that this particular kind of phenomenon or this particular kind of theory, you can just apply directly to mitochondria and Y. Okay, so this is when molecular genetics or molecular biology was making its first inferences about history and prehistory. And when people started looking at, so people first started looking at proteins and looking at differences in species and saw that there is a molecular consistency, there might be a clock. So the mutations are periodic and you can probably scale them. So people thought that maybe now that the technology is also moving towards generating data on nucleotide on DNA sequences, maybe you can use that kind of theory or these kinds of theory to try and infer about human history. So I'll be talking about the history of Homo sapiens sapiens, or anatomically modern humans. AMH is anatomically modern humans, or Homo sapiens sapiens. And when these things were happening, when all of these were being built, there were two theories which were very, very prevalent in deciphering about the history of Homo sapiens sapiens. So one is called a multi-regional hypothesis, another is called a single origin of Homo sapiens. So multi-regional hypothesis would mean that Homo sapiens are different and they arose almost simultaneously at different parts of this world. So that was what is called a multi-regional hypothesis. And that is because, and the people who were arguing in favor of this would say that if you go to different places, like if you go to Europe, if you go to Beijing, if you go to East Asia, you'll find skeletal remains of hominids, of people who are not exactly modern humans, but are very close to modern humans. Like in Europe, you will find Neanderthals. In Beijing, you might have heard about the Beijing man, the Peking man, the Java man, etc., etc. So there are human or more anatomically modern human-like skeleton remains, and people thought that they are probably our ancestors. Yeah, around the same time. I mean, it's not hugely different. So this was something which was observed in different places of the world, and people thought that we are just descendants of regional descendants of these species. Whereas there is another theory, which was the single origin theory, which says that this kind of changes do not simultaneously happen at different places at the same time. It's just extremely low probability even to have the same kind of things which give rise to anatomically modern humans at all regions of the globe, almost simultaneously at the same time. And they would say that it has to have one single origin. It has to have one single origin, and then it has probably replaced everybody else. So Darwin, of course, predates all of this by centuries, but even Darwin had an opinion on this, and Darwin said that it has to be a single origin of humans. And Darwin's argument was pretty simple. Darwin said that you do not see apes outside of Africa, so it has to be Africa. Not only is that it has to be in one single origin, it has to be Africa, because you only see large apes in Africa. Okay, so we have already discussed this, that this is like a schematic cartoon of mitochondria. It's like a circular thing. It's kind of passed on in toto from mothers to offsprings. There are two advantages of using this as well, because there are regions on this where mutations happen pretty rapidly, and there are some regions of this which are very, very stable, which means that you have two kinds of watches. One is, or you have like two kinds of hands on your watch. It's like a second hand which is ticking, so you can also look at it in a small resolution. You can also have an hour hand which gives you a large resolution thing. So there are two kinds of mutations which you know happens on the mitochondria, so this gives you much more advantage. The same was also true for the Y chromosome. Y chromosome also have regions which are very, very stable. You do not observe changes in that very often. Then there are some regions where there are many, many mutations which accumulate pretty rapidly. So good to have these kinds of things because you have both the hour hand and the second hand and the minute hand in the genome. Okay, so this is Alan Wilson. As we mentioned about Alan Wilson, the chimpanzee versus human comparison. This is the first paper which systematically looked into mitochondrial DNA and human evolution. And what they did was they collected 147 individuals from 5 geographic populations all across the world. All right, and Europe, Africa, Asia, Australia, and New Guinea, and they have then looked at the mitochondria region and they have constructed a coalescent tree of this kind. So 2 individuals, if you compare the mitochondria, if you put this like a sequence, if you compare the mitochondria, if you see X number of differences, then you think that these 2 individuals are most likely closely related, right? That's the reason that when it has diverged from this time point in the past, it only had time to accumulate X number of differences. If they have a large number of differences, they are probably sampled from a time or have an ancestor who goes back a long way, because that's the reason that there are many, many differences which have arose. So that's the advantage of having a molecular clock, and that's the advantage of having the mutations sprinkled on them. And when you do that, when you use this and reconstruct this tree, this is the tree that they found. Now, if you, I do not expect you to read all of this, but if you can see that there are these blue circles which are solid, can all of you see the blue circles which are solid? These are individuals who are Africans, all right? The hollow green circles are Asians. The triangular, where are the triangles? Here are the triangles. So these triangles are, so these green triangles, hollow triangles are Australians. These are New Guinea people who are this solid triangles, solid blue triangles, and then the Europeans are squares. Where are the Europeans gone? Here are the Europeans. Some of the Europeans are here. All right. So whatever it is, the take-home message here is that if you look at the Africans, then there is this group of Africans here who everybody, if you consider them, they're like the first differences here. They're like one branch coming out of the TMRCA. And everybody else in the world is another branch. So if you look at this, this is the most recent common ancestor. You hang this by this most recent common ancestor. Everybody else in the world, including some Africans, are on this branch. All known Africans are on this branch, and some of the Africans are also on this branch. And all of them has a common ancestor here. Whereas these individuals who are from the same population, who are kind of neighbors to one another, their mothers, their ancestral mothers go back a long, long time. So when they did this and calibrated this according to the number of mutations mutational difference that you see between individuals, they said that it's about 200,000 years ago. We have all probably started our journey from Africa. So our most recent common ancestors should be somewhere in Africa. It's about 200,000 years ago. So this is the first one which stirred the debate once again and started saying that there is a single origin for all of humanity, or at least as far as the modern humans are concerned, anatomically modern humans are concerned, and it all started about 200,000 years ago.
So what are the paternal lineage? And this is...
Yeah, so yeah, I'll come to the paternal lineage. Maybe I do not have a slide, but I'll talk about the paternal lineage.
Are they around the same time, maternal and the paternal?
The time estimate. The time estimates, yeah, no. It's very similar, but it's not exactly the same. And there are reasons why it's not exactly the same. But what happens subsequent to that is, so this is Spurs data. So when this happened in this is what? This is, I forgot which year. This is 1987. When this happened in 1987, people only could sequence a little bit of mitochondria. So after that, there was a lot of development in the sequencing technology, and there is one region in the mitochondria which is called a hypervariable sequence. And this paper came out a few years later, where they actually did the entire entire hypervariable sequence. And it's that plus many more new samples. And if you look at this, I do not know whether you can see this at all, but it's very similar again. All of these ones which are hollow circles are non-Africans, and all this which have these solid circles are Africans. Oops. No, sorry, just the reverse. All the solid circles are non-Africans, and all these hollow circles are Africans. And if you go back and see that all of these solid circles eventually have common ancestor here, right? Somewhere here is the common ancestor to all of these non-Africans in the world. So these ranges from people from Papua New Guinea, Asians, to people from Europe and Asia and all places in the world, and they eventually coalesce somewhere here. That individual is like a sister to this branch. And from here, you see the different African populations. So these are the Eastern Pygmies, then there are the Yorubans, and so on and so forth, to the Western Pygmies somewhere here. And also, if you look here, there are these Kum can you see this? This is like exclamation kung. If any of you have seen this movie called Gods Must Be Crazy, the Bushmen in Gods Must Be Crazy. So these are the people who are the Bushmen from somewhere in South Central Africa. These people speak a language which have lot of these click sounds, like, so they have a lot of these click sounds, which is like mimicking one of the earliest kind. So it's like, it's not a full-fledged evolved language. It's like mimicking nature to an extent. So they are called the click language, and these are the people. And if you look at them, I mean, they coalesce to their common ancestors, which is way more or far more ancient to the common ancestor to all non-Africans that you see in the world. So all non-Africans in the world, they coalesce somewhere here. Their coalescence is way deeper than that. It goes much more further in time. Just imagine this, a small tribe living somewhere in Africa, and if you look and reconstruct the ancestral trees, that goes farther back compared to people who are sampled from Papua New Guinea and Europe. So if you look at the most recent common ancestor to people from Papua New Guinea and Europe, the mother of them, common ancestral mother, that individual is way more recent compared to the common ancestor of just two individuals who were sampled from the same click-speaking tribe. So this kind of very, very strongly put forward the hypothesis that yes, we all started our journey somewhere in Africa, and it also is coined the term called the African Eve for humanity, and this is taken from the paper, the Nature paper. It says that it's tracing back to the genetic Eve. This is Alan Wilson's lab again. A few years later, in 2000, the technology is developing, and at this point of time, they could actually sequence the entire mitochondria. The tree pretty much tells you the same story. The colors are continuous. So as you see, with the technology developing, with new data coming up, the resolution just becomes clearer and clearer. So if you look into this, these are all individuals who are sampled from different places in Europe. These are all individuals who are sampled from different places in Asia. So they are all coalescing together somewhere to a common ancestor here. These individuals are all coalescing together somewhere to a common ancestor here, and they are distinct branches of the tree. Eventually, all non-Africans in the world coalesce to a common ancestor who's marked by an arrow here. So this is the common ancestor to all non-Africans here. And this vertical line, below this vertical line, are all Africans. So these are Africans, and these Africans have very, very deep branch lengths, and this is like just sampling a few individuals from here. And prior to this time, it's only Africans in the world. And then you reconstruct this tree, and you also put an outgroup here, which is a chimp data. You cannot see this. So this is the time in around 2000 when people have actually sequenced the chimpanzee data, and this bolsters our understanding. The chimpanzees and we have common ancestors, and it also bolsters very, very strongly the fact that Africans are the first place where we all started. So if you look at this, story, and there are which they postulated. So this has a few things, take-home messages. The first one is, of course, that we all started here. We all started in Africa, and there is only a single origin. The second important thing is, which is also extremely, extremely crucial, which is that half of the time we are on this world. Half of the time. So if we are here for a full football match, then the first half was played only in Africa. So this is our connection to Africa is that strong. And when we moved out of Africa, there is only a very small number of individuals who have moved out, which means that the population actually shrunk to, or there is only a very little amount of genetic material which has really moved out of Africa, right? The diversity of the world, in terms of the genetic diversity of the world that exists, that the huge amount of diversity still exists in Africa, and only a few of it we have moved out of this, and that gave rise to all the populations in all of the places in the world. Okay, so yeah, just one second, 2 minutes. So there are many. So these are the early studies. There are many studies after that, plenty of them after that, which are some of them I'm going to also talk about a little bit, which supported this idea, and the data was overwhelming in favor of this. One of them was Y chromosome. I unfortunately do not have a slide showing that. So if you look at the Y chromosome of the world, it pretty much shows you the same picture, exactly the same picture. The only problem was the basic history remains same. The time estimate using the mitochondria was 200,000 years. The time estimate using the Y chromosome was about 150,000 years. So, but the first half, second half story, that remains same. So half of the time we are on this world, even if you use Y chromosome, so the relative understanding of the history of these trees do not change. There was a bottleneck. There was this long period when we were in Africa and we diverged. There was a huge amount of diversity. Then a few people moved out. All of this holds true. It's only on the time estimates which are different. It has also come up from very strange quarters. So this is tuberculosis. So if you look at the tuberculosis pathogen, which is, you might be knowing that tuberculosis, Mycobacterium tuberculosis is something which humans are only affected, right? There is a different kind of tuberculosis for the one which affects the cows. There is a different kind of tuberculosis which affects primates. So for humans, it's an obligate bacteria. And if you sample the mitochondria, if you sample the TB bacteria from all over the world, and if you do a phylogeny of them, that tree looks like somewhere like this. And this is a much more recent thing. If you sample mitochondria from all across the world, and it's color-coded according to the places from where the tuberculosis is collected versus where the places where the mitochondria is collected are this, at the same region. So if this is South and Southeast Asia, this is also South and Southeast Asia. There are only a few bacteria which are collected compared to the number of humans. And if you look at the trees also resemble very similar. So as you can imagine, a lot of our diseases, even infectious diseases, have very similar histories because we are moving out. When we are moving out, we are moving out with our germs as well. So they have a very similar kind of trees. So coevolution also support this. And this is roughly the kind of timelines that we have. It's about, you can debate about the times, but it's about the first 100,000 years that we are here. About 100,000, we started moving out at different directions. Amazing thing was that Europe, you find very strong evidence that Australia was inhabited much earlier than Europe was. So people have moved from here all the way to Australia much before people have moved here, which is actually pretty close to what Africa is today. And this is the part of the world which was conquered later, which is pretty much understandable because people have to travel all the way from here, and this was connected, and then eventually from here to here. All right, so we take a break here at this point, and we'll start from here.